in sex determination in human beings, number of chromosomes are important. Number of chromosomes. Number of chromosomes in humans, human cell is 23 pairs, as we know. So when you are writing sex determination question, children, try to write a flowchart first. Okay, with a neat handwriting. With a neat handwriting, please keep the side heading. Then you should write the flowchart first. Okay, flowchart. So in this flowchart, you should write male and female. These are parents. And gametes, X and Y gametes. Here in males, both are X, no? So 22 plus X gametes, you should write or represent like this. When you fuse these two, XX fusion, here XY fusion, see how the zygote will be. 44 plus XX, girl, and 44 plus XY, boy, offspring. When you are writing explanation part, try to write like this, children. Try to write like this. So these are very, very important points. Don't write repeatedly. Some of the students are writing one point in repeated manner. Don't write like that. Okay, children. See, children, number of chromosomes in human cell is 23 pairs, as we know. Okay. So in 22 pairs are autosomes and one pair is allosomes or sex chromosomes. Okay. So after that, you should write the sex chromosomes are two types, X and Y. In females, it is XX condition. In males, it is XY condition. Okay. After that, you should write if it is a female gamete, it is having only X. If it is a male gamete, either X or Y chromosomes are there. Okay. And then after explanation of these chromosomes, you should write the fusion. When the female gametes are fused with male gametes, the XX or XY. Okay. Try to represent if it is a female gamete X, the male gamete first X. Then what is the resultant baby or zygote? XX condition. Okay. So the resultant baby is good. And female gamete X is fuses with the male gamete Y. The resultant zygote is XY condition. The resultant baby is boy. So you should, after this explanation, you should write the conclusion. So what is your conclusion? So male is the responsible for sex determination of the boy. Okay, children. So this is the way you should write sex determination question. Okay. So total 11 points I given here. So along with the flowchart, try to write like this, children. This is the man you should write. Okay. This is the representation. And next, note. Look at this note, children. Look at this note. Genetically, sex is not determined in some of the organisms. No, look at here. I'm giving here one as a special note. If possible, please make a note, children. In a few reptiles, the temperature at which fertilized eggs are kept determines whether the animals developing in the eggs will be male or female. So, especially in snails, the individual can change sex. It indicates that sex is not genetically determined. This is also one of the answer. So try to make a note in your notebooks, children. This is also important answer for that question. Maybe it is also one of the important question in exam. I hope, please take a screenshot if possible. Okay, next. Look at here, children. Monohybrid cross or Mendel's experiment, it tells about traits may be dominant or recessive. Monohybrid cross, this checkerboard is very, very important. Try to draw the table clearly. Okay, try to write it clearly, children. So this is the way to represent in your paper also. Okay, children, that's why I am giving here. See how I written on the paper, children. See, first you have to write the parent plants. If it is a tall plant, should be mentioned as a capital T, capital T. If it is a pure dwarf plant, we should mention it as small t, small t. Okay. And what are the gametes which you are taking from the parents' children? The gametes, if it is a tall plant, should take capital T. If it is a pure dwarf plant, it should take a small t. If uses with these two gametes, in what are the F1 generation children? As we know, what are the F1 generation resultant condition? All plants are tall plants. Why? Because here the capital T is representing as a dominant trait. Okay, wherever the capital T is there, that is a dominant trait, so that in F1 generation, all plants are tall plants. So what is the ratio of F1 generation children? 1:1, as we know, 1:1. Okay, next. See children, this is the checkerboard. After F1 generation, you should write F2 generation. Okay, children, in F2 generation, we are mentioned here the F1 generation traits. In between the F1 generation traits, what are the resultant conditions? Look at here. See, this is male gametes and these are the female gametes. If you fuse these two gametes, the resultant traits will be capital T is fused with this capital T. The resultant is capital T, and this one is fused with this small t. The resultant condition is capital T, small t. The same way here, capital T, small t. At last, these two fuses, and these are recessive traits, small t, small t. Okay, children. So you have to clearly mention the resultant traits in a checkerboard very easily. Okay, this representation is very, very important. This type of representation you will get 8 of the 8 marks, children. First, you should write this flow chart, this checkerboard. Okay, next. Based on that, you should write the explanation like this. Whatever the explanation, see children how we have to write monohybrid cross explanation here. Mendel did experiments on pea plants. What is the scientific name of pea plant, children? Pison sativum. Pison sativum. See, Mendel selected pure tall plants as well as pure dwarf plant. Based on the flowchart only, I am explaining this one. See, he did cross-pollination between them. First of all, it is a cross-pollination. In F1 generation, what is the result of F1 generation? All plants were tall, isn't it? Then he did self-pollination between F1 generation plants. Okay, after this, in F1 generation and the self-pollination between these two, what is the F2 generation? In F2 generation, 3/4 plants, 75% of plants were tall plants. Okay, 1/4 plants were dwarf. 25% of plants are dwarf plants. So here representing as if it is a capital T in trait or capital T and small t, it should be tall plant. Or if it is a small t condition, it is a dwarf plant. So based on the above results, a single copy of capital T is enough to make the plant tall. So what is our explanation here? If it is a capital T in the trait, it should be a dominant trait. If it is a small t, it is a recessive trait, as we know. So based on the above Mendel experiment, what we have to say, the traits may be dominant or recessive. Based on the monohybrid cross, we should write this traits may be dominant or recessive explanation. Okay, children. So this is the way you should represent the monohybrid cross, children. My expectation, this is the question in your exam for 8 marks. Okay. Now let us see dominant characters and recessive characters are also there, but it is a dihybrid cross. Dihybrid, monohybrid is different. for a dihybrid cross, children. Dihybrid cross. So this is the trait are inherited independently. This is dihybrid cross question, children. In some of the papers, like UTF paper, in UTF paper, this is the main question, children. Maybe this is also one of the 8-mark question in your public exam. So try to prepare this checkerboard clearly, children. Okay. See, in this question, the main representation, previous question, only 2 alleles are there. But in this question, total 4 alleles are there in a single parent. Okay, here we are mentioning 2 characters. That's why we are taking 2 traits. Each trait consisting of 2 alleles, no? So total 4 alleles are there in a single parent. See, these 2 seeds, we are taking a seeds here. First seed is round and green, and second seed wrinkled and yellow. These 2 are parents here. And out of this parent, what are the gametes which you are taking here? In first parent, capital R and capital Y, I mean. So whatever, so you should take a reverse or pure traits or impure traits. The result may be same. Okay, children. So this is the checkerboard. You already practiced this type of questions. So the explanation also very, very important for this question. After flowchart, you should write explanation properly. Okay, try to write 8 to 10 points after flowchart. Okay, children, that is very, very important. That is very, very important. Try to write a minimum 10 points, children, 8 to 10 points. Okay, next. That is about heredity lesson, 3 questions. Now I'm going to representing here experiments in first lesson. First lesson, we have so many questions, but in lesson 1, we have 4 important experiments, children. 4 important experiments are there. Total 4 important experiments are there. 4 important experiments in lesson 1. In lesson 1, important experiments. Important experiment in lesson 1 in my expectation. First one is chlorophyll experiment. Chlorophyll experiment. And second one is carbon dioxide. Importance of carbon dioxide. Or KOH experiment. This is also called KOH experiment. Okay, in this question, what is the importance of KOH, children? KOH is important for it is absorbing carbon dioxide. Okay, that is a key point here. And third important experiment in this lesson is sunlight. Sunlight experiment. Sunlight is necessary for photosynthesis experiment. And fourth experiment, action of saliva. Action of saliva experiment also very, very important, children. Action of saliva experiment. These 4 important experiments. These are very, very important experiments, children. So, I'm already given explanation about action of saliva here. See, in my expectation, these 2 are important this time. Okay, children. We already read these 4 experiments. Repeatedly, we written these 2 experiments, children. So try to read these 2 also. Okay, try to read. Sunlight is necessary for photosynthesis and action of saliva on carbohydrates. These 2 questions also very, very important questions, children. Now let us see how to write experiments in exam. See, especially when you are writing experiments, how we are representing. That is also very, very important, children. Okay, basing on the writing of experiments, you will get 7 to 8 marks. Okay, so whenever you are writing aim, apparatus, procedure, inference, conclusion, or a result, try to keep side headings. Okay, with a separate black paper, black pen or blue pen. Okay, side headings with the black pen, question number, and after that, Roman and question number, then should keep side heading. So what is the aim of a chlorophyll experiment? What is the aim, children? Some of the papers, they given an experimental diagram, they ask 4 questions. What is the aim of that experiment? What is the apparatus of that experiment? What is the observation of that experiment? What is the result of that experiment? Why should we use KOH in that experiment? Like that, they used to ask some questions. Okay, but in previous papers and this recent model papers, they are giving direct questions of experiments. Okay, see, children, in this, what is the aim of this experiment? See, to prove that chlorophyll is essential for photosynthesis. When you are writing apparatus, children, please give numbering, children. Numbering. Okay, how many apparatus or how many materials you are writing, the numbering is very, very important. Numbering. Okay, try to give 1, 2, 3, children. So it is very easy to guess whatever the materials this girl or boy written in the answer. The examiner will correct it. Okay, see, 1, 2, 3, 4, 5 materials are required for chlorophyll experiment. How I written in the procedure? See, children, procedure, total 10 points I gave here, 10 points. See, take a potted plant with variegated leaves, money plant or croton plant. Please mention in brackets. Then keep it in a dark room for 3 days. Why we should keep it in dark room? For destarching. Okay, for destarching, we are keep it in dark room. After that, whenever you are keeping in a dark room, the starch gets used up, as we know. Okay, after destarch process, then the discharged potted plant is using for this experiment. Then what is the 4th point? Then keep the plant in sunlight for 6 hours. Okay, then you have to remove that leaf from the plant and trace the leaf on a paper and mark the green areas for our understanding. And what is the 7th point? This is the common in both starch experiment also. So boil the leaf for a few minutes, then boil the leaf in alcohol and water bath, and slowly the leaf becomes pale white. Okay, so what is the 10th point? Then test the leaf with a solution. After that, observation. Say, children, how I draw this diagram. Don't use green pen, children, in exam, green or red. For your understanding, I'm representing like this. Okay? Okay. So try to draw the diagram neatly, and then you should place it a box here. Okay, after diagrammatic representation, please draw the table like this, children. Box, children, for this diagram. Okay, so observations. Try to write observations also. Very clear observations you have to write. Okay, so green areas of the leaf, if it is a chlorophyll experiment, green areas in a croton plant are turns blue-black. Yes. Why? Because the chlorophyll is trapping the sunlight. It is preparing carbohydrate so that it turns into blue-black color. Other parts do not turn blue-black color, children. So what is the indication for this experiment? That indicates that photosynthesis success only in green areas, chlorophyll containing areas only. So what is the inference for this experiment? Inference is this activity proved that chlorophyll is essential for photosynthesis. Okay, children. And then try to write if having any precautions. What are the precautions which you are taking in for this during experiment? Try to write a minimum two, precautions here. What are the precautions which you are taking for chlorophyll experiment? We are using an organic solution like alcohol, no? So alcohol is highly inflammable, so take care while you're heating alcohol. And that carefully handle the glassware. When you are using glassware like a petri dish, beaker, water bath, so you should carefully handle the glassware. Try to write this type of precautions. So this is the way I should represent chlorophyll experiment along with the diagrams. Now, the second experiment, it is about carbon dioxide is essential for photosynthesis. This is very, very important experiment, children. Last year, this is the 8-mark question in public exam. See how many materials are there. What is the aim of this experiment? To prove that carbon dioxide is essential for photosynthesis. So what are the apparatus which we are using for this experiment? 2 potted plants, 2 bell jars, 2 glass plates, quartz glass, iodine solution, Vaseline, potassium hydroxide solution. In brackets, you should mention KOH. So this is the way you should mention operators with numbering, children. Numbering. So this is the clear representation of materials. After clear explanation of these materials, try to write in procedure minimum 10 to 8 to 10 points. I'm saying here I'm giving 11 points. 11 points. 11 points. See how I'm represented here, diagrams. Bell jar, potted plant, glass plate, and watch glass with KOH solution. So this is the way you should represent the diagrams during the exam. Okay. See, aim, apparatus, procedure, and diagram, and then observation of this experiment. This is the observation, very, very important observation here, children. See, one of the leaf which nearby the KOH solution does not turn blue-black color. Why, children? Because it is having KOH, potassium hydroxide. So what is the function of potassium hydroxide? It absorbs the carbon dioxide. If there is no carbon dioxide, there is no photosynthesis, isn't it? So that it does not turn blue-black. And second point, on other leaf turns blue-black, it doesn't have any KOH. So previously it is having carbon dioxide. By using that carbon dioxide, it is preparing its own food. Okay, it's representing starch granules in the leaf so that it turns blue-black color. What is the inference here? This activity proved that carbon dioxide is essential for photosynthesis. And for this experiment, what is the main precautions? Applying Vaseline without leaving gap. When you are putting bell jar, so you should apply Vaseline, no? So without leaving gaps, you should apply the Vaseline. That is one of the precautions. And second one is carefully handle the glassware. While you are using glassware, any experiment, you should use it to write this type of precautions. Carefully handle the glassware. Okay, now this is the main experiment. I'm expecting saliva experiment also in your paper this time. Action of saliva on starch. Aim to prove saliva acts on starch. What are the apparatus which you are using for this experiment, children? First one is 2 test tubes. Second one, saliva. Third one is starch solution, 1%. And fourth one is iodine solution. Procedure also important for this experiment. So here we are taking 2 test tubes. 2 test tubes. So take 1 ml of starch solution in 2 test tubes. Okay, test tube A and test tube B. Okay, children. Test tube A and test tube B here. This is test tube A and this is test tube B. Okay. See, after start solution in both the test tubes, add 1 ml of saliva to test tube A only. 1 ml of saliva in test tube A only. And both test tube, we have to leave both the test tube undisturbed for 20 to 30 minutes. Then add after 20 to 30 minutes, add few drops of iodine solution to both the test tubes and observe. What is our observations here, children? In test tube A and test tube B, the solution in the test tube B turns blue-black color, but in the test tube A, it does not turn blue-black color. Okay, children. Why? Because saliva converts complex carbohydrates are converted into simple sugar. Isn't it, children? See, in observation, what is the observation here? The solution in the test tube A turns blue-black and test tube A does not turn blue-black. It indicates a saliva axon start so that the simple sugars does not give any color change. Okay, children. So for this experiment, the diagrammatic representation, look at here. This is the way you should represent the diagram, children. See, in the start solution in both the test tubes, after undisturbed saliva in test tube A, then after iodine test, how the color change is there. Try to mention in test tubes also, children. Okay, after iodine test and before iodine test, how it is there. Okay, finally, what is the inference for this activity? This activity proved that saliva acts on starch. Okay, children. And what are the precautions for this saliva experiment? Here, the first precaution is add saliva only to the test tube A, but not in a test tube B. And also, but add iodine solution to both. This is the clear precaution. And here also we are using glassware now, so that same carefully handle the glassware. So this is also one of the important experiment, children. Try to prepare this experiment also. And in one of the UTF paper and government released some of the papers, the carbon dioxide in exhaled air. How can you prove in exhaled air contains carbon dioxide? This is also there, children. Maybe this type of questions also will come. So try to prepare this question also. Okay, for a respiration point of view. These 4 experiments, the first 3 experiments on photosynthesis, and 4th experiment is about nutrition. And I'm representing here the experiment which is related to respiration topic, children. Maybe this is also one of the important questions in your paper. Okay, so what is the aim of this question, children? You are experimenting about carbon dioxide in exhaled air, so that to prove that exhaled air contains more carbon dioxide. For this experiment, we use it to the requirement is 2 test tubes and lime water. It is a bicarbonate solution also. And third important requirement is a syringe or pitchkari, and 4th one is a rubber tube. So for this experiment, what do we need to do? See, procedure. See, children, these are the 2 test tubes. Take some lime water in a test tube and blow air through lime water by using a rubber tube. I'm representing here. See. So note the time, how long it takes for lime water turns milky. When it is having a carbon dioxide, when you are blowing, if it is having carbon dioxide, the lime water slowly changes into milky white. Isn't it, children? That is a clear representation, the exhaled air which is consisting of carbon dioxide. So what is the 4th point here? Now we take the same quantity of lime water in another test tube and pass air through lime water by using syringe or pitchkari. Note the time, how long it takes lime water turns milky. Okay, so this is the diagram for this experiment, children. Once look at this diagram. It's carrying in one test tube and the man blowing in another test tube directly with the help of rubber tube. And basing on these two, what is our observation? Let's see. The lime water turns milky in less time when we blow air through it. Okay, so that it indicates that blowing air or exhaled air contains more carbon dioxide. Isn't it, children? So, what is the inference for this experiment? This activity proves that exhaled air contains more carbon dioxide. Okay, so whenever the carbon dioxide is there, the lime water is turns milky wet. Okay, children. So, what is the precautions for this experiment, children? Do not blow air forcefully in the lime water. Okay, and here also we are using glassware now, so that carefully handle the glassware is one of the precautions. Okay, next. Same, but fermentation experiment. Fermentation experiment. Okay, so fermentation experiment. See, to prove that carbon dioxide released during fermentation. So, for this experiment, we are using test tubes, one whole cork, bent glass tube, yeast, and fruit juice or sugar solution. For this experiment, we need to take some fruit juice or sugar solution in a test tube, add some yeast to it, and close it with a one whole rubber cork. Fit a bent glass tube through rubber cork. After that, dip the free end of the one glass tube into a test tube containing lime water. Same. So that what is our observation? So yeast during respiration, it should release carbon dioxide, as we know. So that the slowly gas bubbles are released from the sugar solution. So whenever the glass tube, the bent glass tube is delivered directly into the next test tube, which contain lime water. So whenever the carbon dioxide passes through bent glass tube, it is delivered to next test tube with the lime water. So that lime water turns milky. So that it indicates gases and what is the released gas here? Carbon dioxide. So that it gets milky white. So what is the inference here? The activity proves that carbon dioxide is released during fermentation. Now write precautions for this experiment. What are the precautions I have to write, children? One end of the bent glass tube must be dipped in the lime water. That is very important. Why? Because it is a gas, no? So try to dip into the lime water. That is important. So here also we are using glassware, so that it should try to carefully handle the glassware. Okay, children. And next. See, this is very, very, very important experiment. Phototropism experiment. This is one of the 8-mark question. So this is maybe this question is will be there in the public exam. See, for phototropism experiment, diagrammatic representation also very, very important. Here, same experiment we used to represent two important tropisms. What are those, children? Phototropism, geotropism also. Okay. What is the aim of this experiment? The proof that shoots show that phototropism and root shows geotropism. Phototropism means the plant respond towards the light. Geotropism, the roots which are grow in gravitational force, towards the gravitational force. Okay, children, where the soil, where the nutrients will be there, so that the roots will be grown in that. Okay, apparatus for this experiment is conical flask, germinating bean seeds, wire mesh, cardboard box, and water. Procedure of the phototropism experiment. Take a conical flask fill it with water, as we know. Take 2 or 3 freshly germinated bean seeds. and keep them on the mouth of the conical flask, by using wire mesh. Keep the conical flask in cardboard box, which is open one side. Okay, children, this is very important. After the arrangement, look at the diagram, children. How I am representing the diagram here. This is the way I should represent the diagram, children. So after the arrangement, near the window as the light pours down the flask. Okay, then leave the arrangement undisturbed 2 to 3 days and observe. Okay, so that the flask, turn the flask so that the shoots are away from the light and the rods towards the light. So, basing on the development of shoot and root development, what we observe after the growth of these two, what is our observation? In the above two observations, shoots grown towards the light, while roots grown away from the light. This is geotropism and phototropism here. Okay. So what is the inference? At last we mentioned, like this activity proves that shoot show phototropism, root shows geotropism. What are the precautions which you are using here, children? What are the precautions you are following here? Ensure that the seeds are germinating properly. Same here also, glassware is there, no? So that carefully handle the glassware you have to write. So these are the important 8-mark questions. Along with that, budding and rhizopus experiments, bread mold experiments also there, children. Those are also very, very important questions in reproduction lessons. Children, try to prepare those two also. Okay. So now I am representing important diagrams. Important diagrams. Look at the diagrams, children. First important diagram is, expecting diagram is human excretory system. Human excretory system. See, when you are drawing the diagram, see the representation of kidneys and the blood vessels, ureter, urinary bladder, and urethra. So after the diagrammatic representation, try to label the parts in one side. Okay? Okay, children? See how I am representing here the parts, labeled parts. See, heading, human excretory system. After diagrammatic representation, we should draw the label parts with the arrow mark, keep the arrow mark. Then you should write the exact correct labeling part there. Correct labeled part. Don't overwrite. Okay, children? So try to write the labeled part clearly without spelling mistakes. Without spelling mistakes. Okay, children, that is very, very important. Okay, so some of the children, they are labeling the parts. It is not exactly in that particular place, children. So try to practice and where the part is. If it is a renal vein, if it is a renal artery, try to mention clearly. Okay. So then ureter, urinary bladder, and urethra. There is a change, difference between ureter, urethra, children. Ureter, which passes the urine from kidney to urinary bladder, children. That is ureter. Okay, ureter is different, urethra is different. Okay, after the labeling parts, try to keep box. Okay, children, keep the box. Inside the box, you should give your representation well. Okay, children, this is the first expected diagram for your exam. And second diagram is nephron. Nephron. What is a nephron's other name? Filtration unit of kidney. Structural and functional unit of the excretory system. Structural and functional unit of excretory system is nephron. Isn't it, children? So see how the nephron represented here. See, children, Bowman capsule, glomerulus, renal tubule is there up to collecting duct. So this is also one of the important diagrams, children. So try to practice this diagram also. This is very, very important. Very easy diagram when you are practice. Preparation and practice are very, very important now. So try to practice today. Try to practice diagrams. Okay, children. In examination also, which question is need diagrams? So try to draw the diagrams for 4 marks and 8 marks questions, children. Okay, if it is necessarily, I mean, the question is important for diagram representation, you should draw the diagram of the related question. Okay, related diagram, you have to draw the particular questions. Okay, next, after nephron, flower structure, LS of flower, LS of flower. Clear representation of pistil and stamen is important. Clear representation. When you are giving a representation, the labeled parts, especially for flower, try to mention the difference between male and female parts in this structure, children. Okay, stigma, style, ovary. Okay, and here anther, filament are male parts. So what are the names of female and male parts? Stigma, style, and stamen, ovary. Okay, not stamen. Stigma, style, and ovary are called pistil. Okay, these parts are comes under the gynoecium. Whereas anther and filament here, we use it to say stamen. These parts are comes under androecium parts. And the remaining parts, those are accessory parts called petals and sepals. Petals and sepals. So this is also one of the important diagram, children. So try to draw this diagram well. Okay, this is very, very important diagram. See, after the diagrammatic representation, try to draw the box for this diagram. Then, in some of the papers, especially in grand test, pre-final, and old papers, and especially UTF papers and action plan, 100 days action plan of government paper, see, this is also one of the important diagram. You used to ask, try to draw fertilization in plants. There is a question, no children, fertilization in plants. So germination of pollen on stigma or fertilization in plants diagram is very, very important diagram. This time, this is also one of the expected diagrams, children. See, for this question, only female parts we are mentioning here. Okay, children, in this female part, how the pollen grains are arranging, how the pollen grains are attached, how pollen grain is grows like with the help of pollen tube, how it is transfer pollen tube and their gametes into the ovule. This representation is very, very important. So try to mention clearly, children, pollen grain, pollen tube. It is a totally female part now, so that should write style, I mean stigma, style, ovary, ovule. Okay, children? So here there is a question, one more question also there, children. In which area we use it to say fruit area, whether it is a ovule or ovary. Ovule is converts into seed, as we know. After fertilization, the ovary becomes into a fruit. This is also one of the 1 mark questions, children. So after the presentation, try to draw box for this question, and the labeled parts only one side. You should mention the labeled parts in one side only. Next important diagram is male reproductive system. Male reproductive system. The representing parts basing on the flow chart. See, when you are reading this question, children, we use it to start this question with testis. No. Testis after vasa differentia. Vasa differentia also. After that, seminal vesicle. Seminal vesicle after prostate gland. And then urethra. and the outer portion penis. This is the way you are giving your explanation notes. That's why you should mention the label parts basing on the explanation. Okay, this is also one of the important diagram. And next important diagram is female reproductive system. Female reproductive system. After neuron diagram, in old papers, before 2 to 3 years before papers, this is the common diagram, children. After neuron, the female reproductive system also common diagram, important diagram. Okay, so maybe this time a female reproductive system, maybe it will come or not, but this is also one of the important diagram. Try to practice this diagram also. So the labeled parts, see, oviduct is also called fallopian tube. After that, ovary. Okay, then uterus, cervix, and then vagina. Cervix, after the vagina. Then human digestive system. Human digestive system. Human digestive system. If it is a difficult paper or an average paper, sometimes they use it to ask this type of questions also, children. They'll give diagram and they use it to ask 4 questions. Okay, so alimentary canal or digestive system, flowchart as we know, parts flowchart. It starts with mouth. Okay, mouth, buccal cavity, esophagus, stomach, small intestine, large intestine. Okay, small intestine, it has 3 parts. As we know, duodenum, jejunum, ileum. Okay, children. So, basing on the flow chart, you have to prepare for this diagram. Accessory units are liver and pancreas. So, the digestion also important. The chemical changes also important, children, here. Anyway, diagram is there, no? Look at here. In mouth, which nutrient digestion takes place, children? Carbohydrates. There is no digestion in esophagus. We know. In stomach area, HCl is there. HCl is secreted by the walls of the stomach, as we know. What is the function of HCl? Also very, very important. Okay, children, it is helpful for to kill the bacteria as well as it is helpful for the protein digestion process. Okay, next one is duodenum in small intestine. In duodenum area, bile juice, based on the bile juice, it is secreted by the liver, and the bile juice is important for emulsification process. Fat digestion takes place in the duodenum area, as we know. And small intestine, different protein digesting enzymes are released here. In stomach area, protein digestion is initiated. In small intestine, carbohydrates, proteins, and fat digestion takes place with the help of proteolytic enzymes like pepsin, trypsin. And fats are digested by the lipase, and carbohydrates are digested by an enzyme called salivary amylase. So these are also important enzyme names. In exam point of view, these questions will come in 1 mark questions. Okay, children, which enzymes digest the protein? They used to ask enzymes. Protein digesting enzymes are pepsin and trypsin. And fats, how fats are digested, children? With the help of bile and lipase. Okay, how carbohydrates are digested, children? With the help of salivary amylase, thiolene enzyme, isn't it? So those are very, very important enzymes for the process of digestion. Okay, this is about digestive system. Now, respiratory system. In respiratory system, diagram also very, very important. In some of the papers, they use it to ask, explain human respiratory system. Human respiratory system. Along with the diagram, use it to explain respiration process, children. This is also one of the important questions for 8 marks. Okay, So here also on flowchart is there. It starts with the nose, nasal cavity, pharynx, larynx, trachea, bronchi, bronchioles, alveoli, and the alveoli exchange the gases into the blood, into these cells. Isn't it, children? This is also very, very important diagram. See how I am representing one side, important parts, flowchart, basing on the air passage. I am labeled the labelings here. And see, rings of cartilage. This is extra here. How I am representing rings of cartilage. And the diaphragm, which is useful for the respiration process, as we know. This is very, very important for inhalation and exhalation process. Inhalation and exhalation process. So human respiratory system diagram also very, very important. Now, next important diagram is human heart. Human heart. See, here also an 8-mark question is there, double circulation. Okay, what are the components of the blood? And try to explain the cardiac cycle. They used to ask like that. This is also important diagram, children. But when you are representing heart diagram, clearly mention the oxygenated parts and deoxygenated parts here, children. Oxygenated and deoxygenated areas. When you are mentioning these two, try to label correct way. In correct way, try to label the parts, children. Instead of right or left, don't get confused. Okay, don't get confused. Right side deoxygenated, left side oxygenated. Okay, basing on that, you should label the parts. Okay, children. See, aorta, pulmonary artery, pulmonary veins, left atrium, left ventricle. These two ventricles are separated by the septum. Now, right side, right ventricle, right atrium. In that right side, there is a superior vena cava and inferior vena cava. So, I'm mentioning here vena cava from upper body, vena cava from lower body, or superior vena cava, inferior vena cava, used to write. Both are correct. Okay, children, this is about heart diagram. So, try to practice this heart diagram also. Today, we should practice these diagrams, children. See, human transportation system. How the double circulation takes place in humans. This is the representation of double circulation. We have double circulation and single circulation differences, no, children. So you should draw the diagram, and it is a 4-mark question. Try to draw the diagrams also for this diagram. I mean, for this question. Okay. Next, very, very important structure, that is neuron or nervous cells. Till some of the students, they get confused when we are giving neuron. They thought that it is a nephron. No. Okay, children. Neuron is also called nervous cell. Neuron is also called nervous cell. Don't get confused. Okay, children. Neuron is different. Nephron is different. Nephron is an excretory filtrating unit. Neuron is a unit which can transmit impulses from brain to target organs. This is a very, very important diagram in the nervous cell. Very, very easy diagram also. Very, very easy diagram. So my expectation, you will get a nervous cell in this exam. Okay, how I am representing, see, children? See, axon. How I am representing axon clearly. One of the hand is extended, that is the axon now, from the cell body. From cell body, we have some branches here. Those are dendrites. And the prominent nucleus is there. Prominent nucleus is present in the diagram. In cell body, it is having prominent nucleus. Then axon. Axon is covered by myelin sheath. The yellow color part is myelin sheath, children. Myelin sheath. Okay. In myelin sheath, there is a gaps are there, no? Gaps. The gaps are called nodes, Ranvier nodes. Those are also called Ranvier nodes. Each myelin sheath, the dots are there, no children. Look at the dots. The dots are nothing but Schwann cells. Okay, and the ending parts is nerve endings, and reflex arc, reflex arc, reflex arc. See, in this reflex arc, which is one of the involuntary action, which is coordinated by the spinal cord. So during reflex action, how the impulses are transmitted from the target organ to the spinal cord, which is mentioned here, and effector is there, sensory neurons are there, and relay neurons are there, and receptor cells, how receive the impulses, how it is relayed in spinal cord. That is a representation is there here, children. So this is also important question for 8 marks. Okay, along with explanation. It is there in our pre-final paper, no? So reflex arc, important diagram, and very, very important diagram. One more important diagram, that is brain. See, children, the clear labelings are important. Forebrain, midbrain, and hindbrain. Forebrain, which is coordinates all voluntary functions. All voluntary functions. Please keep it in your mind. See, in one of the question in previous paper, the bicycle riding is coordinated by which part of the brain they asked. Which part, children? This is the voluntary action. So that it is controlled by cerebrum. Okay, please keep it in your mind. If it is a voluntary action like writing, reading, running, if it is a voluntary action, it is coordinated by cerebrum. Okay, so involuntary actions are there. Those are controlled by the medulla oblongata in some of the actions, in medulla oblongata in some of the actions are cerebellum, okay, hypothalamus, like that. Involuntary actions are controlled by these midbrain and hindbrain parts. Okay, see, children, pineal body, master gland is there in brain, that is pituitary gland. That is pituitary gland. Okay, children, see, cranium, how I represented here? Cranium, cerebrum, midbrain, hypothalamus, pituitary gland, hindbrain, pons medulla, cerebellum, and spinal cord. These are very, very important parts for the diagram. Okay, this is about human brain. Next, nutrition in amoeba. Nutrition in amoeba. This is very, very important diagram. See, in some of the paper, it is like a 1 mark question. In some of the UTF paper, this is the question, it is given as 4 marks. In that 4 marks, you try to represent the diagram and explanation. You have to write explanation also. How food is captured by the amoeba. Okay, nutrition in amoeba. Basing on that explanation, you should draw this type of diagram. Okay, in 4 marks, maybe they used to ask nutrition in amoeba for 4 marks. Diagram or explanation they used to ask. Okay, so how food particle is captured by the amoeba with the help of pseudopodia, how it is forms the food vacuole, and how it captures the food particle. Okay, so this is the representation of nutrition in amoeba. Next, today's session, I want to show some of the questions or 1 mark, which are related to 1 mark. See, important abbreviations. Important abbreviations. Important abbreviations. ATP, ADP, STD, AIDS, DNA, UNEP, CFC. So we can add HIV also here. See, ATP, adenosine triphosphate. ADP, adenosine diphosphate. STD, sexually transmitted diseases. AIDS, acquired immunodeficiency syndrome. And DNA, deoxyribonucleic acid. UNEP, United Nations Environment Program. And CFC, chlorofluorocarbons. So these are the important abbreviations for 1 mark children. And now let us see chapter wise 4 mark questions. Chapter wise 4 marks after diagrams. After 8 marks, some questions I have given. No, so along with the topic, I'm used to give 4 marks and 8 marks now. Okay, so see children, for life process, see, for life process lesson, expected 4 marks. Life process lesson, these are the expected 4 marks questions children. Difference between autotroph and heterotrophs. This is for nutrition. Based on the nutrition, you have to write the differences between autotrophs and heterotrophs. Okay. And the second question, it is about circulation, arteries and veins, differences between arteries and veins. And third one, aerobic and anaerobic respiration. Okay. And fourth one is single circulation and double circulation. And fifth question, xylem and phloem. And sixth one, alveoli and nephron. Maybe alveoli and nephron is the important question, and maybe it is given in, maybe it is the one of the important question for biology exam children. I'm expecting this time. So try to read this question along with these questions. Say seventh one, how is a small intestine designed to absorb digested food? This is also important question. And describe the structure and functions of nephron for 8 marks or 4 marks. Okay, children, this is very, very important. And then life process 8 marks. I'm already explained, but I'm giving here. See, carbon dioxide essential for photosynthesis. Chlorophyll is necessary for photosynthesis. I'm already discussed that saliva on starch, action of saliva on starch. And one more important question is sunlight is necessary for photosynthesis. And to prove that carbon dioxide is released in fermentation. This is also important questions in life process lesson. Now, second topic that is how do organisms reproduce? In my view, these are the important 4 mark questions. See, give suggestions to your friends to aware of reproductive health. Okay, and what precautions will you take to keep away from STD, sexually transmitted diseases? And then third one is about contraceptive devices or contraceptive methods. And fourth one is about slogans of on female feticide. Okay, slogans. And fifth one is how does reproduction help in providing stability to population of species. Okay, with the relevant points you use it to write at least 8 to 9 points here. 8 to, so at least 8 points you have to mention for this question. Then, what are the functions performed by the testes in human beings? And seventh one, what are the advantages of SR over ASR? Okay, next, what is the importance of DNA copying reproduction? Okay, children, next, sexual reproduction and asexual reproductions. Okay, that is. Next, our environment. Our environment, 4-mark questions. Our environment, 4-mark questions. What precautions do you take to reduce ozone layer depletion? Precautions you have to write. And then, what precautions do you suggest to reduce the effect of non-biodegradable waste? We already read this question. These all questions we read. See, third one, suggest some methods to reduce the chemical pesticides. And fourth one, slogans on ozone layer depletion. Next, slogans to make people aware of reduction of chemical pesticides. Okay, and sixth one is what are the trophic levels? Give an example of food chain and state the different trophic levels in it. Okay, and seventh one is about problems caused by non-biodegradable waste that we generated. This is also important question, children. Repeated question. And eighth one is what is ozone? How does it any ecosystem? Okay, this is also important. 4-mark questions here. And heredity. Essay questions. One of the essay questions will come in this chapter, no? So 8 marks. How is sex of child determined in human? Sex determination question. I'm already explained about monohybrid and dihybrid cross, no? So these 2 questions here in 8 marks. Now, control and coordination lesson. Mostly 4 marks. 4 marks like this, children. See, diabetes is a chronic disease. What measures do you suggest to control blood sugar level? Okay, suggestions. And then here, pancreas, which is released the insulin. Basing on that, you use it to suggest some control methods of blood sugar level. Okay, next. Second one is here, the precautions you suggest to take steps in order to avoid goiter disease. Goiter disease, okay, related to thyroid. Okay, next. Third one, involuntary or voluntary or reflex actions. Maybe it is a differences, okay, or individual questions like that they use it to ask. And last one is compare 2 types of movement to show in plants. What are the movements and what are the movements which you use it to write, children? Especially phototropism, geotropism, thigmotropism, hydrotropism. Okay, so those movements you have to explain here. Compare 2 types of movement in the plants. Okay, try to write those movements for this question. So these are the important 4 marks and 8-mark questions of this topic, children. Okay, and in tomorrow's session, this session is for 4 marks and 8-mark questions, and tomorrow's session exclusively for 1 mark and 2 mark questions. Some important 1 mark and 2 mark questions are there, chapter wise. So in next session, in tomorrow's session, see the chapter wise 1 marks and 2 marks. You have to discuss in the next session, children. So that's all for today's session. Today you have to draw the diagrams and try to complete 8 marks and 4 mark questions. Okay, children. So that's all for today's class. So thank you and have a nice day. And please leave the class. Thank you.